We did our presentation on life and leaf litter, and
I'm Leandra Reed.
I'm Abby Honbarger.
I'm Allison Borg.
And I'm Tori Gable.
So to start off, soil fauna. The soil contains complex, diverse organisms in the ecosystem, and it's estimated that there's about between 5 and 8 million different species of these organisms, and their job is to circle nutrients back into the biosphere using decomposition and nitrogen fixation. And some of the major groups present in the community that do these are plants, animals, fungi, and microorganisms.
Some of the organisms found in the soil are nematodes, annelids, and a variety of arthropod species like the wolf spider and some of these other ones that you see. So we did some research on some other experiments that were done by scientists, and they came up with some factors that could affect the population and the distribution of the soil invertebrates. And they said that the amount of soil, the availability of water and other substrates, and the transport of solutes could affect this. Also, according to Wall in Virginia, certain plant species, soil habitat, including moisture, chemical and physical properties, and the climate play a large role in the population size. And finally, dispersal, large-scale disturbance and aggregation could also have an effect on the local species interaction and the diversity.
So another sample experiment that we found was they tested like the actual leaves composing the leaf litter, and what they called sun leaves, which were darker, contained more sugars than what they call shade leaves. So it is seen that more organisms tend to go to the leaves that have more sugars available for them to eat. So larger population size tend to increase with the favorable tree environments.
So let's talk about leaf litter composition. In the American beech tree leaves, usually you'll find springtails, termites, beetles, wettas, ants, and hoppers, and those are the most abundant.
And then with the maple leaf trees, you have wing ants, Diptera, Homoptera, mites, springtails, wolf spiders, isopods, and alchynia. So the objective of our experiment was to determine if the types of nutrients impacts the diversity and the amount of soil in the soil invertebrates.
And our hypothesis was that the old growth would have more abundance and more diversity because due to the fact it's been around a much longer time than we grow.
So for our methods, we first went to the University of Mount Union's Nature Center, and we took walks on both the old-growth forest and the new-growth forest, and we gathered about a gallon-sized bag of loose leaf litter, making sure there was no soil in the mix, just straight leaf litter. And then once we took that leaf litter and we put it in the release funnel to extract the soil invertebrates inside, and as the light shone on the leaf litter, the microorganisms would crawl lower and lower to stay in that nice moist area until they finally fell through the filter into the bottle of alcohol to contain them.
And this is how we collected our samples. We went to the University of Mount Union Nature Center, and first we went to the old growth site where the American beech tree leaf litter is located, and that area is damp and it has less sunlight that comes down to it because of the leaf coverage, and it has a cooler temperature. And then we went to the new growth site, which has the maple leaves, and that area has more sunlight, which in turn gives it a warmer temperature, and it's also damp because of the weather. So here are the results of the American beech tree litter, and to study the biodiversity of the invertebrates, we used the Shanner-Weiner indices. And on the left we have our species that we found, and then N is the number of the species that we found of each, and then the big N is the sample size, and we just added all of those up. And then for the P, we took the N divided by the sample size, and that's the relative frequency. And then we took the log of the P, and then the P times the log of the P.
And these are the results for the maple tree litter. As you can see, the springtails are most abundant, and the total number was 65, but there's more diversity and more different species than in the old growth. and the H max and H prime are also higher in value than in the old growth.
So for our results, we put a nice bar graph together to show that the old growth forest had less diversity but higher populations of the soil invertebrates. So that's in the blue, whereas the new growth had a higher diversity in the orange, but they had smaller amounts of the microorganisms.
So our graphs and calculations showed that there was more diversity of the invertebrates in the new growth forest over the old. However, the old growth had a larger amount of invertebrates present in the samples. The old growth had already been established, so it has had to adjust to the changes in the environment, whereas the new growth has evolved with these changes, so that's probably why there's more diversity. Also, the old growth has less activity compared to the new growth because it's constantly growing, and this is mainly due to the location of fields. So basically just going over everything, due to the fact that the old growth forest has been around longer, there were more amounts of diversity in the rather than in the new growth forest. And our hypothesis was partially supported due to the fact that there was a larger amount of invertebrates in the old growth, but the new growth had the more diversity. And one possibility for the higher rate of diversity is that it's like a new ecosystem is forming, so it's bringing new organisms. These new species are considered pioneers because they are first to occupy the buried ground and may be crowded out as the community develops. And whatever the new forest was previously used for could have also established new populations of organisms. Thank you.